So what I want to do now is to briefly show you some of the aspects of Labster so that you're not frustrated if you've never used Labster simulation before. Within the Labster simulation in Canvas, you're going to hit play. It's going to load, and then once it loads, we're going to hit start. This area here is referred to as your lab pad, and it will give you instructions. The lab pad you can find when you click home. We also have the lab bench here, so I can click outside of the lab pad to see the bench. And there's 3 different areas. We have workbench 1 on the left, we have workbench 2 over here on the right, and then we have workbench 3 here in the middle. If I click on the lab pad, you'll see what it's asking me to do is to look at workbench 3 and click on the newspaper on the workbench and read it. Here on workbench 3, I can click on that newspaper and then I can click on it again to read it. Now to go back to the instructions, I can just hit the back button. Follow the instructions on the lab pad, but it'll take you to different areas. So now it's having us go out to the farm, and we can click on the farmer. We can ask the farmer some questions. So one of the things it's asking us to do here is to click on the double-muscled cows. So we can come in here. I can see this one is a double-muscled bull. Click. And then it might ask you some questions about that trait. Once you've clicked on the double-muscled individuals, it will fill in the pedigree. And I want to show you a little bit about the pedigree. So this is a female and a male. So male over here, female over here. And the line is a mating between them, and these are their offspring. And then we have a mating here, and these are their offspring. What we have is a double-muscled bull here, another double-muscled bull here, and a double-muscled cow over here. This is what a pedigree looks at. In a pedigree, the shaded areas are the trait that you're interested in. In this case, it's double-muscled. And when it's asking you questions, sometimes you want to be able to see other information, like the image. So to answer this question about the pedigree would be really hard if I can't look at the pedigree. So I can come back and I can look at it, and then I can go back to the question. Also, if I need to, I can come over here and click View Theory, and then it'll tell me a little bit about pedigree. So if I'm not clear about what the pedigree is telling me, I can come over to Theory and I can look at that. Then if I want to go back to the questions, I can just click Home again. It'll take me back to that question. So as we move through, one of the things it then asks us to do is to go and collect blood from one of the animals using the syringe in our hand. So we're looking at animal 3-3. So here's the syringe. All we got to do is go collect on animal 3-3. This is this one, so I just click on it. Now it's sending us back to the lab, and we're going to do some lab procedures. And this is one of the main things I wanted to show you because it might be a little bit confusing at first. Now you'll notice it'll give you instructions on the lab pad. It's telling us to put our lab coat on. If we come over here to the lab, we can also see that it's giving us a hint because it's making those blink. So I'm going to click on that, and now we've put on our lab coat. It's telling us to go to workbench 1 to prepare the PCR. So we're going to click on workbench 1. It's showing us the PCR kit.
Tubes from the kit and put them in the rack.
Wanted us to grab the PCR tubes and put them into the tray. So all I did was click there and then bring those over here. Now what it's asking us to do now is to put these different solutions into our PCR tubes. We've got 3 master mixes, A, B, and C, and then we've got blood from 2 different animals, 3 and 7. So we're going to put those mixed together into these PCR tubes. We're going to grab our pipette, automatically put the tip on. We're going to take master mix A. I already took the pipette. I took solution from this one and put it in here. Now I'm going to take solution from this one and I'll put it in here. I will take solution from here and I'll put it in here. And I'm going to do the same thing for the back 3. Take from the first one, put it here. Take from the second one, put it here. Mix C, put it here. So I have the mix A, B, and C in the front row and A, B, and C in the back row. Now what I want to do is to put the extracted DNA. I'm going to put the DNA from this animal into here. I'm going to take another sample I'm going to put it in the middle tube. You'll see that a lot of times you'll do a few of them and then it will do the rest. So what it wants us to do is to put these in the PCR machine, which is going to amplify the DNA. So we open it and we will put them in. So I'm just clicking on them and putting them over here. Then you have to close it and you can start it.
Great.
So after you answer some questions about the PCR finishes running and you can open it. So it's going to move these PCR tubes over to the rack. So once you load a few, they'll do the rest. And then it'll tell you to start the electrophoresis. So we'll hit start. Once you take a look at the animation, it runs the gel, and then it also determines the size of the different fragments, and that's what it's adding into this pedigree now. So now that you've seen how to click through the different areas within the simulation, I think you should be able to complete that simulation. If you have any questions, please let me know. And as mentioned before, all of your work that you do in this Labster simulation, your quizzes that you take while you're in the simulation, all of that information will come directly over to Canvas. So you don't have to save and send anything. It will automatically come into Canvas into your grades.